And the ball spins and spins. And yeah, we are commencing here. So let's go back to, I've said all we really have time for about Mendel's laws. But let's see if we can get a little bit more on what exactly those things called genes are. Now, Mendel had shown that genes are not not like blending, that genes are some kind of little particles or elements, as he called them, some sort of little things rattling around inside a cell. But it would be nice if we could get a better picture as to what those genes actually are. Are they really marble-like things inside a cell, or could they be something else? Well, we've got to backtrack a little bit and go back all the way to the 1600s, when there's a London gentleman, really brilliant guy, by the way, one of the most widely, what's the word I'm looking for? He designed buildings, and he did medical experiments, and he did physics experiments, and he wrote music. One of the great all-rounders of all time, a man by the name of Robert Hooke. And he was trying out a new invention of his called a compound microscope. Now, magnifying glasses were already known, but Hooke had the idea that if you put a couple of magnifying lenses in line with each other, you could magnify things much more highly than you could with a single lens. So he essentially invented the compound microscope, although it looked a little bit different from the one that we use now. And he's the one who looked at a drawing of a slice of cork and published this drawing in 1665. And he noticed that it was made up of a whole bunch of little chambers. This is the reason why cork floats. It's very light because it's made up of all of these little air-filled chambers. And the chambers reminded him of the cells in a monastery. I don't think this is what he had in mind, but we also use the same word to mean, you know, the cells in a prison. So he called these chambers cells. We actually talked about this a little bit when I was talking about plant anatomy. Many functional tissues in a plant are actually dead and have to die in order to be functional. And the bark of certain oak trees, actually all trees would have some of this, but it's especially thick in the bark of a particular oak species called the corkbark oak. No surprise there. That bark can actually be peeled off and harvested and used to make corks in wine bottles and things like that. And it is dead tissue. The actual living parts of the cells all died a long time ago, but what's left is those cell walls, which make up these little chambers in a slice of cork. Okay, Hooke drew a whole lot of things that he'd seen under the microscope, and it just blew people away. Nobody had ever seen fleas and ants up close before. And people had also been wondering for a long time about the problem of heredity. Human sperm cells were discovered in 1677. It was fairly obvious to almost everybody that sex had something to do with babies, but exactly how the trick was accomplished, nobody quite knew. And in 1677, a Dutch guy by the name of Leeuwenhoek, who'd invented a different type of microscope, actually discovered these. It took egg cells quite a bit longer to be discovered. Eggs were first seen in 1797. Because egg cells are internal and there's only one that's mature at any given time. At least in humans, it's that way usually. And, you know, they stay inside the body, whereas sperm cells are produced in great numbers and it's quite a bit easier to get some for study. One early idea was that either eggs or possibly sperm cells, like this diagram that you see right there, Actually, that each cell contained tiny duplicates of the parent. So every sperm cell consists of a teeny tiny, 10 micron little baby folded up inside the head. And that this would implant itself in the mother's uterus and just kind of inflate into an embryo and then into a baby and ultimately into an adult. And then how does the next generation get there? Well, that folded up baby in the sperm cell must have countless millions of folded up babies inside the baby's own sperm cells. And then those must all have folded up babies inside their sperm cells and so on. So basically, the entirety of humanity is nested one generation inside the next, like so many Russian dolls. This also means, of course, that if every sperm cell contains a baby, then your average teenager kills about 100 million babies every day, sometimes twice a day. Let's not get into that too much. Suffice it to say that the development of better microscopes showed that this is basically a false hypothesis. There are not tiny folded up babies inside sperm cells, just so we're clear on that. What is in there is worked out in 1839 by a couple of Germans named Matthias Schleiden and Theodor Schwann. Again, I might test you on last names, but no more than that. You don't have to know birth dates, death dates, or even first names. And Schwann had been looking at animal tissues. Schleiden was a botanist looking at plants. And they found that when they looked at plant and animal tissues in their, improved microscopes, both of them seemed to be made of a limited number of different types of subunit. And these were the cells that Robert Hooke had found. And Schwann and Schleiden proposed that all living things had to be made up of cells. So these are some drawings that they made of cells that they saw. They originally stated that a cell was defined by having a nucleus, and we now know that's not true for prokaryotes. Leave that aside for right now. We now know there's a big exception to the rule that all cells have nuclei. And as Schleiden put it, though the variety in the external structure of plants is great, their internal structure is simple. This extraordinary range in form is due only to a variation in the fitting together of elementary structures, which indeed are subject to modification, but are essentially identical. That is, they are cells. Much like, all of the buildings on the UCA campus have different shapes, different sizes, and different functions, but they're made out of a fairly limited parts list. They're made of the same types of brick and the same types of glass and the same electrical fittings and things like that. They're made out of a relatively limited number of possible parts. And what makes them different is not so much the parts, it's the way in which the parts are put together. Same thing here. Plants can range from, grasses less than an inch high all the way to 300-foot sequoia trees. And they can range from, I don't know, lily flowers to cactus to who knows what. But what makes them different is the way in which cells are put together. And there's a relatively small number of cell types that a plant is made of. All right, we're letting somebody in here. Hello, welcome, welcome and bienvenue, welcome to cabaret, en cabaret. Right. So where was I? Right. Plants are made up of a fairly limited number of cell types that can be combined to make anything from a tiny grass to a sequoia. Mammals range from tiny mice to blue whales. And, armadillos to people, there's not a great deal of difference in the cells that make us up. There's not a huge difference in the cells of you compared to the cells of an armadillo. You could find some differences, but it's not very big. The difference between you and an armadillo is not so much your cells, it's how the cells are arranged, how they're put together, how many of them there are, how they're connected with each other. Schleiden and Schwann originally thought that cells might kind of form sort of droplets and kind of precipitate out or crystallize out from some kind of living fluid. But after further study, a gent by the name of Virchow stated that cells could only come from other cells. Omnis cellula e cellula. All cells come from cells. So how do cells make other cells? By the 1870s, better microscopes were making it easier to see things inside cells. That 1840s microscope is fairly simple. By the 1870s, that microscope is just about as good as the one that we'd use in labs today, at least optically. And something else that might not seem relevant at the time is that the artificial dye industry had taken off. A British guy by the name of William Perkin, don't worry about his name, but he'd accidentally discovered a compound in a waste product of the gas industry called coal tar. And he discovered a compound that happened to be brilliant purple while analyzing coal tar in 1856. And his purple dye, by the way, was called mauve, M-A-U-V-E, and was extremely popular for about a decade. It was the kind of thing that, queens would dress themselves in. This is a mauve dress from about that time. That was the first of what became a flood of artificial dyes that got used in the fabric and clothing industry. Before that, dyes were all derived from plants. There was a fairly limited number of shades, and they tended to be expensive. People who weren't well off generally wore white and brown, the colors of undyed cloth. Now, of course, you can get wild colors and just about any shade you can think of. You can get a t-shirt in that color, and that ultimately stems from the discovery of mauve purple in 1856, followed up by an awful lot of new colors that were being found. And the reason I'm even bringing this up is that some of those artificial dyes turned out to be very useful if you added some to cells and looked at them under the microscope. Dyes increase the contrast. A lot of cells are pretty much transparent. There are some tricks with the light that you can use to see detail and bring out detail in a transparent cell. But dyes, or when we're talking about biology, we usually call them stains. Stains will not only increase contrast, but some stains will stain certain structures, but not others. So they might stain the nucleus very strongly, but not the rest of the cell. They might stain some types of organelle, but not others. There's a whole bunch of these. If any of you are in applied health and you take a histology course, you might actually get to play with them at some point. I'm sure you've heard of something called a pap smear. It's a test for cervical cancer. Not a great deal of fun to have one, I'm told. But in a pap smear, you take cells from a woman's cervix and you hit them with this cocktail of stains that is made up in such a way is that it will make cells that look like they could be cancerous stain a different color from healthy cells. Kind of a neat trick there. So staining is very important in medicine. And using this, in 1873, a guy by the name of Schneider became the first to observe something odd about the nuclei of cells. He was actually looking at cells in the tail of, I believe, a salamander tadpole, a baby salamander as it was growing. And as it was growing, the cells were, growing and dividing as well. And he noticed something odd about the nucleus. Normally, a cell nucleus looked like a round, grainy blob, kind of what you see over there on the far left. That's a cell that is just hanging out and not dividing, not making more cells. It's just living its cellular life and talking on its cell phone and whatever. But as a cell divided, the nucleus transformed into a set of little string-like things. And the string-like things would actually move in some kind of pattern. Second from the left, you can see these string-like things that have moved into the midline of the cell. And then third from the left, you can see how those string-like things seem to be pulling away from each other. And at the right, you can see there's now two clusters of little string-like things as the entire cell is starting to divide. And these little stringy things do this kind of dance where they appear, move to the center, and then pull apart every time a eukaryotic cell divides. Prokaryotes, it's different. Prokaryotes don't have nuclei, and they don't have these stringy things. But maybe we'll talk about that much later. But certainly all eukaryotes have these stringy things. And when a cell divides, there can be modifications to this, but this is the basic process that the nucleus goes through. The nucleus turns into stringies. The stringies line up in the middle. The stringies separate and pull apart. Now, we can't just keep calling these things stringies. These objects happen to be very strongly colored by many stains. That is a cell that's been stained with, I think it's probably a stain called methylene blue. And you can see that most of the cell is a very light pink color, but the stringy things in the dividing cell stain a lot more strongly purple. And these objects came to be called colored bodies, or in Greek, they came to be called chromosomes, a term you've probably heard before. So there you've got a cell that is about to divide, and you have a bunch of chromosomes inside, colored, stringy things. Right. Very careful study with the microscope showed that when a cell began to divide and the chromosomes were visible, each chromosome would be made up of two parallel strands. Now, the strands are parallel and they're so close to each other that it's not always easy to see this. Going back to this picture, if you look very carefully at the stringy things, you might be able to see that they look kind of double. That they are actually two colored strands with a slightly lighter zone in the middle. It's not easy to spot. You have to be, you have to be careful with this. You have to look really hard, and it probably helps if you're on drugs. But each chromosome is made up of two parallel strands, and we call those strands chromatids. So these are chromosomes taken with a much more sophisticated type of microscope called a scanning electron microscope. We don't really have time to get into how that works. But this is two chromosomes, and each one is made up of one strand, there, and a second strand, there. So there's your chromosome. Each one's made of one strand and a second strand. And those two parallel strands are called chromatids. Oh, I haven't figured out how to make the boxes go diagonal, but you can see that the second strand, the other chromosome to the right, is made up of two strands as well. And incidentally, the two strands meet at a meeting spot that's called the centromere. So there we've got the chromatids. And the two chromatids are connected at only one spot. There's not more than one, at a spot called the centromere. Mere is just Latin for a division. Centro means in the center. So the centromere just means the central division, the central part of the chromosome. All right. Here's the thing. When a cell is not dividing, you can't see the chromosomes. The nucleus just kind of looks like a grainy blob, and you really don't get much detail. But they do seem to be present all the time. Chromosomes are always there, even when you can't see them. And one demonstration of this is the fact that the number of chromosomes stays the same throughout all generations. If you have a cell with 10 chromosomes and you let it divide, and then you look at it, you look at the two daughter cells afterwards, they will have 10 chromosomes. And the cells that come from them will have 10 chromosomes, and so on. That number stays the same from one generation to the next. I mean, occasionally it will change, but that's usually an error. That usually means something's gone wrong. And the number of chromosomes is actually characteristic of a species. These are human chromosomes that have been stained with a new type of stain that fluoresces under the right wavelengths of light. So if this picture looks a little bit kind of neon-y, that's why. And don't worry right now about those numbers. What you need to know is that almost all cells in a normal human have 46 chromosomes per cell. If I was to take one of your cells right now and squish it on a slide and do a chromosome spread, I would find almost certainly 46 chromosomes. That's typical for humans everywhere. If I did the same trick with a fruit fly, I'd get 8. If I did the same trick with a horse, I think I'd get 62. If I did it with a mouse, I'd get 40. The number is just a number. There's nothing really interesting about the number, except for the fact that it is characteristic of species. Almost all cells of all members of a particular species will have the same number of chromosomes, and that number stays the same as a cell divides, and divides again. The motions that chromosomes go through when a cell divides is called mitosis, as in, I should have worn warm socks because now my toes is cold. And imagining all of you laughing uproariously the way that live students would do in happier times. And I'm going to show you some pictures here, and then we're going to go and look at this live. Well, they're prepared slides, but I'm at least going to do some live microscopy at the end of this. These cells, by the way, that you're seeing happen to be cells from embryonic whitefish. Embryos have lots of cells that are actively dividing because that's how the embryo grows. When a cell is not dividing, the nucleus just kind of looks grainy. Now, the chromosomes are there, but they're not visible. So this is a cell that is not in the process of dividing. It's just kind of hanging out and doing whatever cellular things it needs to do. When the cell begins to divide, usually, there are some exceptions, the membrane that separates the nucleus from the rest of the cell, called the nuclear membrane, usually will disappear, and the chromosomes start becoming visible. We will get to later into how it is that they can go from being invisible to being visible. Let's just say that right now the chromosomes are starting to clump together and get large enough that you can see them. So that is a cell in the process of, the chromosome becoming visible. This is a cell that's embarked on the first stage of division, and we call that stage prophase. And yes, you got to know all the stages. Interphase, prophase is when the chromosomes become visible. And while this is happening, we now know that there are two tiny little organelles called centrosomes that produce a set of microscopic fibers inside the cell. The fibers are called spindle fibers, and the structure is called the mitotic spindle. They're hard to see, not impossible, but tricky with ordinary microscopy. Today, we have techniques that can take images in which certain structures in a cell are fluorescent. So this is a picture of a cell in prophase with the spindle fibers stained fluorescent green and the chromosomes in blue. The chromosomes are still kind of this mass in the center. And then to the left and the right, you have what look like octopus with entirely too many tentacles reaching in to grab the chromosomes. Those are actually the spindle fibers making up this thing called the mitotic spindle. And at the far left and far right, you can't really see it as a separate thing, but that's where the centrosomes are located. The centrosomes are organizing the formation of the mitotic spindle fibers. What you need to know right now is that the spindle fibers are kind of the motors. These are what are actually going to make the chromosomes move. Chromosomes can't move on their own. They don't have muscles or anything like that. For division to take place, they have to be dragged around by the spindle fibers. So in prophase is when they form. In the next stage, the chromosomes line up in the center along an imaginary plane called the metaphase plate. The metaphase plate doesn't really exist. The metaphase plate is just an imaginary plane cutting the cell in two that the chromosomes line up in. And this is what metaphase looks like. If I didn't make it clear, these are stained cells. Chromosomes don't really look dark blue, but you can see them in the center right there. That central clump of kind of rope-like things are the chromosomes. And in this one, you can actually see just a little bit of the spindle fibers. They're still pretty faint, but those two dots that are stained blue at roughly your 2 o'clock and your 8 o'clock positions, or maybe your 2:30 and your 7:30, to the lower left and to the upper right, those dots are actually the centrosomes that are organizing the spindle fibers. And they're kind of surrounded by this halo of very faint pink spindle fibers. So you can barely see them here. This picture shows them a little bit more clearly. You can see the spindle fibers are glowing bright green in this case. And the spindle fibers are attached onto the chromosomes, which is what makes up that blue blob that you're seeing right there in the middle. Then comes anaphase. And in anaphase, the chromosomes break apart at the centromeres. And these separate chromatids migrate to opposite sides of the cell. Remember that when cell division started, each chromosome was made up of two chromatids stuck together in the middle. Now's the point where the two chromatids separate from each other. That centromere breaks, and separated chromatids are pulled to opposite sides of the cell. And again, it's the spindle fibers that are doing the pulling. These are the little motors that are moving them. So in this picture, you can see we've got two clumps of chromosomes that are being pulled away from each other. That's what it looks like under fluorescence microscopy. You can see those two light blue blobs are chromosomes, and they're getting pulled by the fibers of the mitotic spindle. So the spindle fibers are in bright green. And then at the end of it all, we call it telophase. And in telophase, chromosomes now clump up into new nuclei. New membranes form around them, and the chromosomes lose their individuality. They seemingly kind of dissolve into the grainy stuff that you saw at the very beginning when the cell was in prophase. New nuclear membranes form, so you end up with a cell with two nuclei. And usually, the cell itself will pinch in two at the same time, and that's a process we call cytokinesis. If this doesn't happen, you end up with a cell with two nuclei, or sometimes with more. There are cases where that does happen. We don't really need to get into there's different reasons for that. We don't really need to get into when and why that happens. This is not something that you would see in humans. But usually the whole cell will pinch in two, and you now have two separate cells. And the process of the whole cell dividing up is called cytokinesis. And usually cytokinesis and telophase happen at the same time. They're not the same thing because the phases I've given you, interphase, prophase, metaphase, anaphase, telophase, IPMAT, those phases are technically apply only to the nucleus, not the entire cell. But usually at about the time of telophase is when the entire cell will pinch in two. So usually cytokinesis happens at the same time that telophase does. And there's what this looks like under fluorescent microscopy, where we've got two completely separated bundles of chromosomes in blue pulled apart by the spindle fibers. I should mention that plant cells do something a little different in cytokinesis, because plant cells are surrounded by a rigid cell wall. They can't simply pinch apart. What will happen in plants is that a new cell wall, which you can faintly see as a very lightly staining greenish line beginning to form in the middle of the cell, that new cell wall forms to divide the cell in two. It's a little bit like, if you wanted to divide up a room in your house, you'd probably build a wall partitioning that room off into two. We've actually done that in Lewis Science Center with offices and classrooms and things like that. Every so often they get partitioned. And this is what plant cells do. Building that wall is the way a plant cell does cytokinesis. Now, I could keep going, but what I'm going to do right now is plug in my microscope camera, and this will take me just a moment. There we go. Okay, I'm going to assume that everybody can see this. I'm going to start by looking at some animal cells that have been stained and killed in the process of undergoing mitosis. And these happen to be cells from the developing embryos of an intestinal roundworm. So yeah, kind of yucky, I suppose. But this is actually a pretty good way to see what mitosis actually looks like. And let me see, I'm going to kind of zoom around a bit and see if I can spot okay, kind of right there in the center, you can see a cell that is ending telophase. You can just see in the middle of those two cells that they both have chromosomes that are still visible. Those are those slightly darker areas. I'm going to fiddle with the lighting to see if I can make this more clear. Probably not. And then cytokinesis has also happened in that cell has almost completely pinched into two. So that in the middle right there would be a cell in late telophase. It's almost, mitosis in that one is almost over with, and we just have two cells now. Let me see. What else we got? Okay, let me see. At the far right, we've got another one that's in telophase over there on the far right. Just below center, we've got a couple that look to me like they're in prophase. Like especially, let me see if I can get, yeah, there's one right there in the center, right there. You can really see the chromosomes there. They're those dark blue thready things right there. That one is probably in prophase because it doesn't look to me like they're organized in any particular way. In a cell, if a cell is in prophase, you will see individual chromosomes, but they won't have any particular order to them. They'll just, be kind of flopping around, tangled up like a plate of spaghetti. So that would be a prophase right there in the middle. Okay, I'm gonna noodle around and look for a couple more, and then I'm gonna show you what this looks like in plants. Here we go. Oh, here's a good one. Okay, let me try to get this guy. Yeah, the one closest to the center right there now, that's a nice anaphase. You can see two bundles of chromosomes at about the 12 and the 6 that are caught in the act of pulling away from each other. Above and below, you can see a little bit of kind of brownish fuzzy stuff. That's actually the spindle fibers. I'm going to try slightly higher magnification. Sometimes I get kind of disappointing results if I do that with this camera. But let me see what I can come up with here. Okay, that right there is just two cells. But where was that nice one? Okay, right. Okay, right there. Yeah, there's some dust in the system that I've got to clean off. But right there, you can see the cell still looks sort of, yeah, okay, there. I can make some of that go away just by messing with the lighting. Yeah, so that's a cell that is in anaphase right there. You can see the two bundles of chromosomes as those dark, slightly bluish areas, and you can even see the bunches of spindle fibers above and below. So that's a cell that got trapped in the middle of anaphase. Let me see, right over here. Right over here, we've got one that's in late telophase. You can see that the two bundles of chromosomes have been pulled apart, and you can see how the cell is starting to pinch into two. So that's telophase with cytokinesis. And then let me see. Yeah, there was one over here. Not that one. Is it that one? Yeah. Okay, that one looks to me like metaphase, because to the left and the right, I can see two bundles of spindle fibers, and in the midline, kind of running vertically down the cell, I can see some of the chromosomes. I don't think that's the full number that it's got, maybe because this is a slice through the cell. But there we've got the chromosomes lined up in the middle. So the metaphase plate is this kind of imaginary plane that's dividing the cell in two. So I've shown you telophase, metaphase. Let me see if I can find a decent prophase, because, you know, why not? We're here, we might as well. Okay, what's this? Okay, this is probably another metaphase, because there you can really see the chromosomes clearly. I can't really see spindle fibers clearly here, so this might be a late prophase, but they could also be sort of lining up in the middle. So I would call this late prophase to early meta. And let me see if I can find one more prophase, because, you know what? Then I'll switch slides. Is that one? Okay, yeah. The one right here I'd call early prophase. You can just see these little kind of bluish blobby things in the middle there. There's not really a well-defined nucleus. As I focus up and down, you can see the little bluish blobs in there. Those are chromosomes that are just coming to be visible. So I'd say that's an early prophase. So I think I've shown you what all of the stages looks like. And I'm going to switch over to the tip of a plant root. And let's try to get that in focus. Okay, that's not it. That's the edge of the cover slip. Okay, where is this darn thing? There we go. Okay, the funny thing about plants is that they grow at the tips. And you might recall that I mentioned this in lecture. I'm going to go to the lowest power for just a second. And what we're looking at right there is the very tip of an onion root. Now, if you buy onions at the store, you don't normally see the actual root because they're cut off. The onion that you eat is actually, believe it or not, it's technically swollen leaves. The layers of an onion are thickened leaves wrapped tightly around each other. And the actual roots are, if you've ever pulled an onion out of the ground, you'll have seen little, you know, kind of long thread-like things sticking out of one end. That's the actual root. Now, the way plants grow, in most plants, all of the cell division, all of the making of new plant cells is taking place at the tips. You have to go farther back down the root. The cells that have been made are solidifying their cell walls, and eventually they get a full cell wall and they can't expand anymore. That's when the cells are mature. But the actual cell division is taking place in these zones at the tips of the roots and the tips of the shoots that are called meristems. You might remember that word from way back. So this is a section through an onion root meristem. The cell walls have all been stained green, and the nuclei and chromosomes have been stained purple. And you can already see there's some interesting little purple blobs in here. But let's go ahead and get a closer look real quick. All right, let me see if I can invoke a higher power. There we go. Okay, so here's a bunch of plant root cells. Most of them are in interphase. You can see that, okay, the cell walls and the cytoplasm, that's the gooey stuff inside the cell, is green. The nuclei are stained pink, and in most of these, you can't really see any detail inside the nuclei. The nuclei are just these grainy things. But if we try to center it up here, okay, closest to the center right now, I've got a nice example of, I'm going to fiddle with the light a little bit, but right there in the center, I've got a very nice example of telophase. Sorry, I lost it. Sorry, working with the microscope makes the controls start moving in the microscope controls move in the wrong direction from what I'm used to. But let me see, I'm going to center up on, or try to, on that cell that's in telophase, because there's actually a number that are surrounding it that are worth looking at. Okay, there. As close to the center I can get is an onion root cell that's in telophase. You can see there's two lumps of chromosomes that are stained pink. In between the two, you can just barely see a faint, slightly darker green line. That's the new cell wall coming into existence. So that's a late telophase. Directly above it, there's actually a cell in late anaphase. You can see, or maybe early telophase. You can see two separated lumps of chromosomes that have been pulled completely apart. To the upper left from the cell we started with is actually one in prophase. You know, in interphase, the nuclei are kind of this faint pink. But at the one that you're seeing at the upper left, you should be able to see that the chromosomes are much more colorful. And you can barely see that they're starting to become these separate entities, but we can't really count how many there are yet. That they're still kind of, tangled together like spaghetti noodles on a plate. And then to the right of the one that we started with, we're probably getting one in metaphase, where the chromosomes are kind of lining up in the middle, along the midline. So this one that's in the center now, that would be a metaphase. There's probably another metaphase up and to the right of it. And then this is the one, not that one. Well, actually, that one's a little better right there. And I've just lost that one too. Sorry, gang. Okay, there's one right there that's a really nice prophase just above center. You can see the chromosomes. They're like little strands of yarn, I guess, but they're still kind of jumbled together, and it's hard to count how many there are. So in prophase, the chromosomes are visible, but they're not especially organized. This, hang on. Yeah, nerds. Okay, this one over at the right, that would be a metaphase. The chromosomes are visible and they're actually kind of lined up in the middle, although the chromatids are kind of sticking out to either side. But that's what an early metaphase would look like, lined up in the middle. I'll find just a couple more and then I'll take a break. But I did want to at least let you see what this looks like, even if I can't give you all microscopes. So that's one just a bit above center right now that is probably prophase or maybe early metaphase. You can see the individual chromosomes pretty clearly. And let's see, Okay, here's a good. There we go. There's a nice anaphase closest to the center right there. You can really see those separate bundles of chromosomes that are being pulled apart. So that's an anaphase that's closest to the center. And just to the left of it, you can see a prophase that's really nice. The chromosomes are now starting to get, really clearly visible right there. There's a prophase, and kind of cut off right now, but there's that's probably a late prophase to meta. Oh, here's a good meta. I'll show you a good meta, and then I'll give you a break. Right there, that's a good metaphase. You can see the cell wall, the little box that it came in. The chromatids are all sticking to left and right, but if you look carefully, you can see that the middles of the chromosomes are all lined up in the center. They're all lined up on a line that runs from approximately your 7 o'clock to your 2 o'clock. That's where the centromeres are, and then the chromatids are kind of sticking out to the side, but that's as nice a metaphase as you're likely to see. That's a gorgeous metaphase. That's a metaphase any cell would be proud of. And again, most of the cells that are surrounding it, you're not seeing individual chromosomes. There's just this kind of grainy pink stuff in there. That's what interphase looks like. I'll try to remember to post it, but you can actually find time-lapse photos of mitosis as it's taking place. You can watch this in real time. Typically, it would last about an hour, maybe a little more, maybe, but an hour is about right. But yeah, there's some nice time-lapse photos that will let you trace this. But you can also just look at stain slides like this and just see what the stages are, and then work out, what comes before what, and work out the stages of mitosis. Okay, right. Okay, I'm going to go ahead and stop recording right there, and I've got 11, so we can come back at 11:15. I'll give you a nice long break there.